So we have a comprehensive strategy to address that question because we realize that cytokine release syndrome and neurotoxicity are important side effects that are linked to cell therapy usage. So for that, we are exploring multiple approaches in clinical research. One of them is the earlier utilization of steroids at an earlier grades of CRS and neurotoxicity. We're looking also at prophylactic usage of the steroids, as well as combination with some novel therapies like anakinra, siltuximab, for example. So what's exciting is that we're starting to observe some good signals from the preliminary data of the early corticosteroid usage that we've employed in one of our registration studies. And the data show a reduction in the rates of the grade 3 adverse events for both CRS and neurotoxicity. This is preliminary results that we are encouraged by and would like to see the data mature, and we'll hopefully be reporting the full analysis in a future medical conference soon. So obviously we observed that not all patients would respond to the current approach for CAR T therapy with CD19 specific target. So that's why it's going to be important to find approaches to help increase the efficacy and also mitigate the relapse of patients who initially respond and then they relapse again. And for that, there are some interesting approaches to do it. One of them is the dual target approach. So instead of only targeting CD19, also targeting CD20. So we have our plan that's in place to really explore bispecific and bicistronic constructs that address the two targets. Because what we've observed from research is that those patients who relapse and they lose CD19, majority of them would still maintain CD20, so it's still a very important target to address. So on top of that, we would also like to have multiple targets that are relevant to the different disease indications, and CD20 is another important and relevant target that could be instituted in a sequence with another anti-CD19 CAR T. Importantly as well is that we want to make that therapy more widely available. We try to make it available much faster, and that's why we explore our opportunities with the allogeneic approach. And what that means is that we take the cells from healthy donors, we go through different manufacturing steps, and then make some lots available for patients who are needing this therapy, really available off the shelf. Once those patients are identified as eligible for the treatment, they can receive it, and you cut the duration that the patients have to wait while the full manufacturing process takes place. It's also very important to bring that therapy to patients with solid tumors. We know that really this is the majority of cancer patients. We know that 80% of metastatic solid tumor patients eventually succumb to the disease. So that's a space of clear, high unmet medical need. So we're also exploring opportunities to develop TCRs in that setting. We have a number of programs that are ongoing. MAGE A3, A6 is one of them. HPV E7 is another one of them. So these are targets that are expressed by a number of solid tumors, including disease areas like non-small cell lung cancer, cervical cancer, and bladder cancer. And we hope that we generate some data that would support further development in that space. We also have some collaborative efforts with some research institutions and with the NCI exploring neoantigen approach, for example, which I think would be the true pinnacle of personalized therapy moving forward, and it's really a part of the very big excitement about the future.